So dear students, I welcome you all. Today we are going to see about botany. So most of you have been familiar with the botany courses. So today we are going to see about the A to Z of botany, right? So now everyone understands what is botany, right? So B-O-T-A-N-Y, right? So botany. Mostly botany deals with the plants, various plants, right from the algae to angiosperms. So today present day plants are also known as angiosperms, right? So today we are going to see what and all we are having inside the botany courses, right? So after your 12th standard you would be in a bit confusion stage, what to choose. Then most of the students will be thinking about, okay, if you choose botany, whether it would be successful, whether we would form a successful career in this course. Well, the answer is yes. So mostly there are a lot of courses available after your 12th standard, right? Beginning from biochemistry, biophysics, then microbiology. But there is something unique with your botany courses, right? So after your 12th standard, when you enter into a college, for example, say BSc botany course, so you will be having the following subjects, right? So you will be dealing with algae. It is also known as phycology. Then fungi. It's also known as mycology. Then anatomy. Embryology. Taxonomy. So, well, coming to phycology part, so algae. Well, algae, most of we are familiar with algae, right? So the algae, the person who studies algae is known as algaologist, right? So algae, in Tamil it is known as pasi. So mostly kadar pasigal, or there are a lot of algae available, freshwater and marine. So interesting to know that many of you are familiar with the particular algae, right? It's known as spirulina, right? Spirulina. So this particular algae has been very famous among the nutritionists. So past 2 decades, this spirulina has been going around in and around world literally because it has more antioxidant properties. Antioxidant properties, right? So in simple terms, to make you understand, it deals with anti-aging, right? Okay. So spirulina cultivation, it has been in large scale nowadays in India. We will find everyone doing this spirulina cultivation. So even we can do it in our own lab, right? It's a very simple process. Most of the algae we can cultivate inside our lab. Simple. We can take a spirulina culture, cultivate inside a bubble top. You have the mineral water cans, right? 20 liters capacity can. You can grow an algae inside it also, right? So why we are taking algae, right? Algae is a starting plant. So evolution of plants, when we see the evolution of plants, right from the algae, the first formed plant was algae, or in other words, it's also known as cyanobacteria, right? Cyanobacteria. So from this algae, next there was a shift. It was known as bryophytes. Then there was a third group which was known as pteridophytes. Then came the gymnosperms. So from gymnosperms, there arose a new group called angiosperms, right? So from algae to bryophytes, bryophytes to pteridophytes, pteridophytes to gymnosperms, and then angiosperms. So algae, the algae was, till now we see algae only inside the water bodies, right? So they wholly depend on water. Next came the bryophytes. these are also known as non-vascular cryptogams. These are the first land plants, but although they are land plants, they require most of the percentage of water. Then these are the pteridophytes. So pteridophytes usually they are also land plants, but here we call it as vascular cryptogams, right? Because there is a whole lot of difference from bryophytes to pteridophytes because there is a shift. So this was depending on the water. Now pteridophytes, they also need water, but for certain purposes. Otherwise, they can survive inside in a terrestrial area. So pteridophytes, from the pteridophyte part of view, you will see the plants having their well developed structures like root, stem, leaves, but not exactly root, stem, leaves, but we can differentiate, right. In algae and bryophytes, we call it as thallus, right. So this, from algae and bryophytes, we call it as thallus or thallophytes. So because they bear a structure called thallus, they are known as thallophytes. Similar way, in pteridophytes, they have a unique structure. So there is a differentiation into root, stem and leaves. So this is the precursor of the present day plants, right. So then there was a slight variation, and from pteridophytes there arises a new group called gymnosperms. In this we have a well developed reproductive structure. From this part of plant area, we will be seeing the plants developing iron, vascular bundles, other striking features came across from this gymnosperms part. Then the present day angiosperms we all know. For example, if you want to know the mango tree, right, it belongs to angiosperms. So now we see a whole lot of difference from algae to angiosperms. So it took so much years to evolve among themselves. So this, in the same way, we are going to, when you enter a college, you will be seeing all these courses, right. It comes under botany. So from now a short glimpse of algae, right. So algae we have seen, and most famous scientist is Fritsch. So he has classified the algae into various classes. So like for example, if you take green algae, Chlorophyceae, then red algae, Rhodophyceae, like that, brown algae. So there are a lot of algae forms available. So there is a present relevance to algae, right. Mostly we will be hearing to the terms called bioremediation, right. So bio, the term bio means right. So this is a term which have been listening to all the worldwide. So whatever problem arises, the moment we get into biological remedy, right. So whenever you encounter a term, you should say, you should break it up, right. So any phrase you receive, you should not take it as it is, you should break it up. So you should, there are two parts, bio and remediation. So it's a remedy, remedy means solution, right. So what is solution? Based on the biological means. So biological means are usually, they are not carcinogenic, they are good, nature friendly, right. So bioremediation, so life, bio means life. So to taking the part of algae, right. So bioremediation, when you visit some industries, right, there is an industry called Perfetti. So you will be knowing about the chocolate, famous chocolate called Alpenliebe. So the company which makes Alpenliebe is Perfetti, right. So these industries like this, there are many industries, I am just quoting an example. So these kind of industries, what they do, they develop large content of chocolates, right. Then after the development of chocolates, there will be vessels, they have the content, they do not have vessels like we have it at homes, right. So it is not easy to clean up. So for that purpose, they need a solution for this, right. So mostly what they do, they depend on algae. So the algal scientist or algologist, what they do, they have a proposal, what they do, they go to that particular industries, they know what algae can grow in that particular area, and they develop such algae, make it grow. Then the algae, what it does, it grows inside that conditions, right, the wastewater, it grows under the wastewater and removes the waste, and the water would be reusable. They will reuse it for other purposes. So why am I telling you about this? Because algae is a plant. So from plant, when you can do a bioremediation part, then how successful it would be, right. it would be a cheaper alternative to other wastage. So it's easier to make a waste management purpose. So mostly like algae, there are many more plants. So when we depend on the natural resources like plants, we would be in a position to make it environmental friendly. In the similar way, if we go for remediation using chemicals and all, there are chances of carcinogens, right? Carcinogens, you means the chemicals which are harmful to the society, right? Like waste management, like when we do nuclear reactors, there are a lot of energy waste. So we are not able to cope up with the waste management. So in that purposes, we will, when we use an alternate source like plants, we would be in a better position to make it, right? So algae. So the bioremediation of algae, which is the research is going on, right? So many of the scientists will be, if you visit their websites, when you start browsing about these things, you will get a lot of things. So next comes the mycology. So everybody knows about the fungi, right? From the school days would be surprising to know about fungi. So it's not the particular fungi is not an alien, right? We are very much knowing about the fungus. So fungus, the moment when you say fungus, we are reminded of bread, right? So when we see at homes, we buy a bread packet and we keep it as it is, right? When it attracts some moisture, after a few days, when we see a bread, there would be some sort of green spots, green, black and all, right? These are all fungus. So usually in schools, the teachers would give you an experiment. Just go, if you want to see a fungi, just go take a bread piece, sprinkle some water, keep it aside. After a few days, you will notice some patches, right? This is the fungus. The first fungus which we see during the school days, right? These are all fungus. So the fungus, there are a lot of fungus. Again, there are a lot of classifications available fungi. So mostly in UG, they deal with the classification of Alexopoulos. So the Alexopoulos is the person who classified many fungal organisms inside the criteria. So the subject which deals with mycology, right? The subject which deals with fungus is known as mycology. The scientist who studies mycology is known as mycologist. The same way, phycologist. So fungus. So among the fungus, I would like to share an interesting feature of a fungi. It's a saprophytic fungi. You know the term saprophyte. Saprophyte means dead. So the organism which lives on the dead matter is known as saprophytes. So there is a similar saprophyte, right? The fungus name is Pylobolus. So the fungus name is Pylobolus, right? So that is a striking feature for the Pylobolus. So this is usually grows in dung, right? Dung fungi. So it is also known as dung fungi. So if you want to notice this Pylobolus, you can do it directly. So again, this is also a lively demo, right? So if you take a fresh cow dung, right? If you take a fresh cow dung, keep it inside a Petri plates, right? So there is a dish called Petri dish or Petri plate. So this is a Petri plate. So the scientist who discovered this is known as Petri. So after that the scientist, it was named as Petri dish. So this is a Petri dish usually used in labs. So if you take this Petri dish, place some cow dung, right? Then sprinkle some water and close the lid. After a day or so, you will find certain fungus that would have grown out of this. So when we see it directly, we would not be able to identify because it's a very small thing, right? So when we enlarge it, then would we will able to see the structures. So what would happen, right? When we open the lid, so upper part of the lid, there would be some spots. These are all the spores. So which would have arise from the fungus part. So the fungus would look like this. This would be the stem part, stalk, or so the upper part there will be a portion, right? This is called as spore, right? This is a spore which gets stuck on the upper side of the plate. So usually if you want to view it, you use a electron micrograph, right? Electron microscope images, we will be able to see it in a better way. So apart from that, this is a thing, okay. So the pilobolus thing, it just, the spore just gets stuck into the upper part of the petri dish, right. This is an interesting feature of a dung fungi, pilobolus. So it comes under the group called zygomycetes. So this is the same fungi group in which also deals with the bread fungi, right, that is Rhizopus. So right, so in this way these are interesting features of fungi. Apart from that, these fungus are also marine, right. Not only we see this fungus in terrestrial habitat, we are also seeing is aquatic fungi. There are many aquatic fungi, there are many disease causing fungi. So again, this is the saprophytic part. Then it comes, when it comes to the disease part also, right, because fungi we name it as a plant, but they are also grouped under microorganisms. They are not classified under microorganism, but we call it as a microorganism, right. So the fungus has a dual part. Usually it has a plant part, that it as a microbe it can cause disease, right. So disease part, if you see Candida, there is a fungus called Candida. So there are a lot of species under which comes under Candida. These are fungus which causes many diseases, right. So there are positive and as well as negative aspects of fungi. Then anatomy part. So this mostly plant anatomy, right. So in this course you will study about the plant sectionings. So usually there are sections like stem. So when you take a plant part, for example stem, so when you take thin sections, right, there will be a sectioning. There is itself, the sectioning part itself is a greater task. So you need a lot of patience for this. So when you take a section like this, you will be easily note this down, what are all present in the stem structure. So this is called as internal part, right. So there are, again, for sectioning part, when we take it, there are two sectionings available, right. In short, it is known as TS and LS. TS means transverse section, then LS means longitudinal section. Right. So when we take a TS, right, so we keep a stem like this, we use a pith. So when we keep the stem, we take a section, right. After taking a section, what we do, we will place a thin slice. So to make you better understand, it just imagine that you are taking a potato, then you are chipping out. When you see the chips part, right, in the similar way it would lie, but it would be in more thin, right. The more the section is thin, the more clarity you get. So when the section is thin, when you observe it, then there is a lot of process after this. Then you have to mount it. You will use a slide, right, you will use a glass slide. You will keep this, before that what we will do, this you will take this section, you will dip it in a staining solution. So mostly we use safranin stain, right, safranin. It's some sort of pinkish color. So it is a stain called pink color. Why we use stain? To highlight the areas, right, because when we take the section, when we observe it directly, we would not be able to differentiate between the various parts, right. So all will look like green and all. So to make it differentiate, we use safranin stain. So when we dip the section in the safranin stain and then we mount it in a slide, right. When we mount in a slide, then we use a cover slip. There is a round structure, okay, it's made up of glass or plastic. So there is this thing is called as cover slip. We keep it on this specimen, right. Then before that we use a glycerin, right. We use glycerin. We will fill it up, all areas, because to keep the specimen from drying, right. Otherwise the specimen, if the specimen dries out, we will not be able to watch it clearly. So before that we add a glycerin and we keep a cover slip on it. Then after that, when we observe under the microscope, you would like to know that every regions, right, Whatever and all you will see in the stem part, you will be able to view it. But again, this sectioning involves more patience, right? Usually what the students do is, they take a section, they would see certain regions would be good enough and other regions would not be visible. So this arises when the sectioning is not it's not clearly taken. The students would be taking a section like this, some part would be thin, some part would be thick. So we should avoid, when you get a good section, it should be thin on all sides, it should be universally thin, right. So like that there are many specimens available. When this is an art, again this is an art of taking a sectioning, right. Then embryology. So embryology part, usually we deal with embryos, right, monocot, dicot, these and all comes under angiosperms mostly. So these, you know about embryos, there are a lot of globular, heart shaped and these kind of embryos, the study of this things comes under embryology. Then the taxonomy part. So mostly this is the toughest part, I would say, because tough means it needs a lot of books, right. So taxonomy part, dealing a taxonomy part is not that easy. So taxonomy part when we take, so mostly plant taxonomy, right. So plant taxonomy deals with the naming of plants. So what we see, for example, if you take a mango tree, right, the scientific name is Mangifera indica. So like that, there are a lot of names, right. So scientific names we give, but how would we say that this belongs to this group or this plant belongs to that group? So for that there is a separate textbook or book, okay, that is known as Gamble. So there are a lot of volumes available for Gamble, it's an old book, right. So usually what do they say, what the taxonomist, so the person who studies taxonomy is known as taxonomist, right. So what the taxonomist does, he takes this book, he will observe the plant, right. For example, first he will say the leaves, he will study the leaf part, he will say okay, if how the leaf is arranged, in what direction and all, then he will see that particular area inside the book, then he will say okay, this plant has this kind of leaf, right. Next he will go to the flower part, like that from top to bottom he will analyze all the things, and after that final conclusion he will arrive, okay, this plant belongs to this genus or this species. Like that he will do all sort of research and he will come to the conclusion of the plants, right. So this is a really tiresome job. So this is not easy, because similar two plants would look similar, certain small characters would be differentiate among them. So we have to be very cautious while doing this work, right. So these are the major part you will study. Apart from that, you will be having some electives and all. In electives you will mostly study about microbiology and all things. So apart from this, again let me get into the general part of the botany. So these are the courses we are studying, but apart from that everybody will be having in mind, right, what would be, so by studying botany what I gain, right, and what is the purpose of this. You will say this, these are all things we know, sir, okay, then what is the purpose of studying botany, right. So job part, before that we will enter about the medicinal plants, right. So medicinal plants, so this term has been echoing in our ears for more than 100 years or something, right. So medicinal plants, everybody will say medicinal plants, right. So there are some medicinal plants pertaining to India, right, like Acalypha indica, right. In Tamil it is known as Kuppemeni. So you would find this plant anywhere. So it's not a plant which is rare to see. You will see this plant everywhere in and around your areas, but we don't use it properly. There are a lot of medicinal properties inside this plant, right, but we fail to do this because of lack of knowledge, right. in this way, again mostly in South India, South Indian families, everyone knows about neem, right? Neem plant. It is also known as Azadirachta indica. Right. So Azadirachta indica. So this neem has been among the Indian households, right? Everybody would be using this plant. So it's also related to religious purpose, as well as there are more antiviral and other antifungal properties inside this plant, right? So, but we fail to do so, right? That's why the foreigners, what they did, they wanted a patent right for this. There are many plants, many Indian origin plants, and which we fail to utilize it, right? So neem, so good antiviral property, it has a good antiviral property. Then there is another plant, in Tamil it is known as so other plants like this, there are a lot of plants, okay. So based on the medicinal plants, what we do? So based on the medicinal plants, there are a lot of therapies available, right? Treatments, so medical treatments. So first we take about the Indian treatment, that is called as Ayurveda, Siddha and Unani, right? So all the three medical treatment facilities, they use these medicinal plants. So when we take Ayurveda, they usually take the whole plant, while in Siddha they use the bark or root region. They concentrate on this part more. And Unani, it's a mix up of both, right? So you would be having in mind, sir, why should I take Ayurveda or Siddha? There are a lot of career prospects in this. So if a student does not get a neat seat, then there will be a question, I want to become a doctor, but I am not able to do so. Then what are the options? You have option. You can be an Ayurvedic, right? There are a lot of institutes available. You can apply for an Ayurveda. You can become an Ayurvedic practitioner. Then in the similar way, Siddha also. These are all supported by the government. So you have a bright future in these areas. So for the career prospect, you would be in a good position. Then research part, right? So the medicinal plants, you have this, but what about the research? You have a good scope of research in these plants. Mostly what they do, they take these plants, they do the analysis. So mostly they use some solvents like ethanol and methanol, right? They use these solvents and they go for phytochemistry studies. Right. So this is a major part. Most of them are using these techniques and they are going for research. There are other parts also available, right? So medicinal plants, there are another region called as endophytes, right? So these are the microorganisms which resides inside the plants, right? So usually these kind of microorganisms are not harmful. So they just reside inside these plants. So there are a lot of research going on this also. So some scientists or some researchers, they take the plant, they do the research part. But some, they choose these endophytes which are growing inside the plants and they go for the similar studies, or they concentrate on other medicinal properties using these endophytes, because these grow inside the plants, right? So when the plant is able to inhabit this type of compounds, so why don't they do? So just to grab the attention of these endophytes, right? Then there are similar things available inside this research areas. So the research part, it's not an easy task, right? So it takes a lot of things. So when we take a research in a botany course, you will be easy to the main merits, okay, for merits of using merits of botany in research. Right. So when a student would be having in mind that if I take a botany, whether I am safe in research, yes, because you would be getting ample number of plants, right? You will not be restricted to any particular kind of thing, right? So this is the main reason for choosing a botany course for research. You will be getting a lot of plants, okay. And another medicine plant which I left, so like Unani, Siddha and Ayurveda, there is a medicine called back remedy, right. So mostly you will not be familiar with this name, but everybody knows flower medicine, right. So this is again, this is all buzzing around now, the flower medicine. So whenever you see some persons, they will be having some pills. So this is a flower extract. So these are all flowers which have a anti-stress properties and all kind of diseases. So this is not restricted to particular thing. So the flower medicine when you take, they deal with all kind of stress management, they deal with some diseases, so these are flowers. So the back remedy, they are known as back remedy, this is a foreign concern, right. So what they do, they concentrate on this particular kind of flowers and they go for the medicine properties. So again, this also comes under botany, right. So another subject which I wanted to tell you is, using this botany course, there are certain things available, okay. So modification. So usually in agriculture part, so if you visit some villages, you will see some wild plants, right. There are some wild plants, but they would be some, the characters would be, they would be more disease resistant, right, disease resistant, but they don't produce much grains. They would be disease resistant, but they would produce less grains, right. So to make these plants, to how to overcome this situation, so there evolved some a subject called plant breeding, right. So for example, when we take two plants, right, A and B, right. So A part would be having disease resistance, then B plant would be having more yield capacity, right. So we want both these characters, then what we should do? We would combine these two plants, but practically it's not possible. So there are certain techniques like emasculation, right. In this what we do, so to avoid the self pollination of a particular plant, they would do this emasculation technique, right, removing the male part. So what they do, they remove the male part so that the other side of plant, right, the male part from the other plant would be easily reachable. So in this way we can mix up different characters. So in the similar way, this is a conventional method or oldest method, plant breeding, because it requires more work. So in the same thing, now there is a field called biotechnology, right. In biotechnology, that too in plant biotechnology, to overcome this situation, they use various techniques called plant tissue culture and all, right. In this way they make the work easier. So now we are going to see about biotechnology, right. Again, this is also a buzzing word. So in this there are animal biotech as well as plant biotech, but now I am just going to relate you this to the plant breeding technique. So while we are seeing that plant breeding, when we use a plant breeding, we have certain restrictions. It is a more tiresome work. So when we use biotechnology, we would reduce further time, right. So in this using a biotechnology thing, we would be seeing about a term called, or a bacteria, right. It is called as Bt. In short, it is called as Bt. The whole name is called Bacillus thuringiensis, right. So this is the bacteria which was introduced into plants, right. So we would be hearing these terms called GMO, right, genetically modified crops. So what is genetically modified? So based on the biotechnology concepts, right, we use this bacteria. So this was the case, how they developed a plant, Bt cotton, Bt brinjal, right. While, so if you take the case of Bt cotton, right, so what happens is this bacteria is inserted into this plant. So when the bacteria is inserted into this cotton plant, what happens? The whole plant has this bacteria. So when a pest or any insect tries to digest these leaves, the moment it starts digesting the leaf, it dies. So what happens inside this? The gene, okay, the bacterial gene goes inside the insect, it kills the insect, right, immediately. It enters the gut and it immediately kills it. So for this purpose, so okay, why we concentrate on this, why the Bt cotton was introduced? Because there was a problem. Most the cotton plants, right, when they are early in generation, right, when they start blooming the cotton. So during this case, when pest or insects affects it, the plant would die or the plant would not able to develop further. To avoid this situation, what they did, they introduced this gene to prevent the insect or pest from creating any problems to the plant. But they got a good result, right, they were able to defend or prevent the pest. But the same cotton, when we consume, when we use for our purpose, right, because cotton is mostly used in dressings and mostly useful for humans, right. So there was a question inside every mind, right, okay, the bacteria would kill the pest, but what about the human beings? What would happen if we use this same cotton again? So because of this, this has become a burning issue. In the similar way, the vegetables, okay. So usually you would be familiar with the Bt brinjal, okay, then leave it aside. So just go to a market, see the tomatoes, right. Previous years or 5 to 10 years back, you would see that the tomatoes, when you buy a good tomato, right, what would happen? It will spoil within 2 days. But nowadays, we see the tomatoes, they don't get spoiled even after a week. So there should be something inside it. So we should think. So this is the part where bio comes inside. So mostly we say, okay, we have a tomato, doesn't get spoiled, okay, it's a great relief for us, we will keep it aside in the fridge for more than a week. No, there is something fishy. These are all genes that plays inside the tomatoes that makes them keep it longer. So on long run, that would cause some damage to the human. So we should keep it in mind. So we should not think about the economic part, right? if you buy a 1 kg tomato, dump it in a fridge, then after we use after 1 month or no, it's not at all good for health. So in that case, don't go for the artificially synthesized plants. Okay, use raw materials, freshly what we get, tomatoes or any vegetable in the pot. So go for the natural ones. Okay, then again the bio term. So I just wanted to share some more experience about the bio part. So bio toilets. So mostly you would have heard the term called bio toilets. So these are the bio toilets which use some bacteria to degrade or to take out the waste. These are all implemented in Indian Railways. So if you visit a toilet, see, just look for details. You would find that there would be details called bio toilets, right? In the similar way, bio remediation, which I told you regarding the algae, the similar way bio remediation also used with microorganisms. So you will be hearing about the concepts called oil spill and all. So in that case, they use certain microorganisms to degrade the oils and to make free the water. So in that way, from A to Z, whatever we use, if you include plants or any other biosource, we would get a better life. Thank you.